My name is Ursula Heins, and I am researcher at the University Medical Center of Utrecht. I recently joined the Batten group, but actually I was working on Batten disease since a while. I did my PhD on CNL3 and CNL12 disease in Luxembourg, and this year I moved to Utrecht to continue my work on CNL3. I started to work on Batten disease because I was very interested on rare neurodegenerative disorders, especially affecting children. I found that whatever contribution can really make a difference. So it's the reason I took this challenge back in 2015, and I decided to stay and continue my work on this field. So what I was mainly doing was disease modeling. So we were trying to recapitulate the disease in simple models. We were using yeast, Batten yeast, no zebrafish, patient lines, and the idea was to study what was going wrong in the cells, and then try to identify potential therapeutic targets. Specifically for CNL3, we developed a zebrafish model in which we remove the CNL3 gene using a technology called CRISPR-Cas9. And for our surprise, actually, the fish do not show any symptoms, so it's not so lethal like in humans, potentially because the fish doesn't live so long like human does. But what was interesting is when we look more deep in detail is that all the metabolic abnormalities that was reported in patients could be observed in the fish. And we are not working actually with adult fish. We are working with the progeny, so with the larva stage, so only with fish that are still 5 days of life. And this is what because they are quite tiny, are transparent on that age. Also, zebrafish has external fertilization, and so this allowed to see process under the microscope. And we see that was very impressive to see how abnormal accumulation of these metabolites were observed in so small animals. And this will help us now to try to screen for drugs that potentially can bring to a normal state to the metabolism, and correct these abnormalities, and then the best drugs will be tested in more complex models. So why we are using zebrafish, as I already mentioned, and is a great model because of the size and the transparency, but also is much more cheaper comparing to other animal models. It is quite versatile. It is easy to manipulate, to do genetic manipulations, like removing a gene or putting a gene on top, so we can create lines in which the neurons is fluorescent, or the mitochondria fluorescent, or the lysosome is fluorescent, and this allows to speed up in our studies. Also, as well as they are transparent, we can see the brain under the microscope, something that is not possible in humans. So zebrafish is not only great for aquariums, but also for biomedical research. And during the years, I will have been in a strong connection with the Batten Disease foundations, mainly with the German one, but I need to say it's a quite valuable work that they were doing to try to connect the researcher among us. So sometimes we are not so many working on this rare disorder. So sometimes we lack the contact. So this is the great job that the NCL Foundation was doing, is try to organize meeting and support a lot the young researchers to interact with each other. And I think by now we have a really nice small community that we regularly meet in this Batten Disease meeting. And I think this really helped to foster collaborations and to speed up the research.